Okay, I have posted the next homework, so I will talk about it for just a second here to introduce it, and then we'll keep talking today about genome-wide association studies. All right, here's homework 5. Excuse me. It is due a week from today, so you don't have quite as much time as you've had on some of the other ones. It's not all that long, though. There's a package in R, bioconductor package, that will meet today or tomorrow called Snips, and it is all about doing GWAS. So you have, like we've talked about, you have thousands and thousands of SNP markers across a genome, and we're going to sequence each of those markers. And often we'll have, or we will always have, some kind of response variable of interest, which could be categorical or could be quantitative. If it's categorical, like cancer or not cancer, say, then what we could do is do basically logistic regression. Logistic regression, yes or no, cancer or not, in terms of genotype for the SNP. So is SNP 422, is it a, let's say, a CC, a C on both chromosomes for this individual, or is it a C and a T, or a T and a C, or a T and a T? So we could do logistic regression, log odds of cancer, modeled in terms of genotype, and then since we're doing regression, we could include other covariates like gender and age and race and other stuff. The response variable could alternatively be numerical, in which case we would just do linear regression. Mean response, which could be blood pressure or PSA level or whatever we're trying to model, written in terms of genotype and gender and age and race and all that stuff. So Snips does this for you. If you provide it with genotype information, it will give you a p-value for each of your SNPs. So typically we would have in a GWAS study, we would have a bunch of people, say, some of them have a disease and some of them don't, and for each one of them we obtain one of these assays that gives us genotype information for each of the 500,000 to 1 million SNPs. Okay. So that's the idea. You're going to use Snips to do a GWAS analysis, and for each of, actually we're just going to look at one SNP in this homework problem. Typically you would have, some number of individuals, and for each individual you'd have hundreds of thousands of SNPs. And you would want to ask for each one of the SNPs, are you associated with the response variable? Just like we've done with differential expression analysis, is the mean abundance of this gene different comparing treatment to control, cancer to normal? Now we're going to be asking, is there an association? Is there a difference in my response, which in this homework problem is categorical? Is there a difference in yes, no, in terms of a SNP? And we would go through and do that for all million SNPs. So we would get 1 million p-values, and we know what to do with a bunch of p-values. We can convert them into false discovery rates and try to pull out the subset of the features, in this case SNPs, that are significantly associated with the response. For the purposes of the assignment, so Snips, this package, will automatically give you a p-value for all million p-value or SNPs in your data set. What I want you to do is just look at one SNP. So go do this association analysis for the first SNP. All that is logistic regression. So you're going to use the association function, which comes from SNPAssoc, to do this, but you're also going to match it manually using R's built-in logistic regression function. You should be able to match it exactly. Okay. Another topic that's going to come up today or Wednesday is this idea of equilibrium, Hardy-Weinberg equilibrium. It's a genetics concept, and it describes a kind of a status quo of how a population creates offspring. So, for example, we'll talk about this more soon, but if I am green eyes and my wife is brown eyes, there's a probability that our child is going to be green eyes. There's a probability that they're going to be brown eyes and other things like that. So if everything is normal, there's no unusual pressures on the breeding and generation of progeny, then we could predict the frequencies of blue eyes and green eyes and things like that. If this equilibrium holds, then, or let me say this, if this does not hold, then it can be informative to us for a couple of reasons. If this equilibrium does not hold, it can be indicative of a few things. One, it could be indicative of sequencing errors. If there's 60% brown eyes and 40% green eyes in the population, we think, but in our actual sample, it doesn't look like it's consistent with that breakdown. We have concern that this equilibrium might not be holding, or maybe there's sequencing errors. Another way that this can be violated, this equilibrium, is if there's selection going on. So natural selection is the process of certain phenotypes having some advantage over others, and so those phenotypes are more likely to succeed, to live, to breed, to pass on their genes. If that is actively going on, then we would not have this equilibrium either. So, what does this have to do with GWAS? You usually will be assuming that this holds when you do GWAS. So we are going to assume that for SNP1 in this case, SNP1 is under equilibrium. It is being parsed out to progeny in a predictable way. There's not selection issues going on, there's not substantial genotyping errors going on, and a variety of other things that could be going on are not happening. So, when you do GWAS, like when you use the association function, you're going to get back as part of the output a p-value that is for testing this equilibrium. Does it look like this SNP is under equilibrium, yes or no? If it's not, then you want to think a little more about that SNP. There might be a problem with that SNP, or it might tell you something else. So I want you to know what Hardy-Weinberg equilibrium is, and I want you to match the p-value. So I'll show you the formula for computing the p-value. I think it's informative to understand what the rationale is behind it. Helps you to, I think, understand the genetics a little bit. So we will talk about that, and then this last problem is tied to part B. They're both equilibrium-based questions. Okay. I posted a couple of things. There's an announcement here that points to some short videos. These are 5-minute videos. There's 4 of them on the Khan Academy website. So Khan Academy, if you don't know about it, is a teaching website that was created by an MIT graduate, real smart guy, who makes these short little videos on everything. Everything from history to math to genetics. There you can learn lots of things, and he teaches very understandably simply. So it's a good place to go to learn about a variety of things. I gave you a link to this page, and there's 4 videos. So some of you could lecture to me about this stuff. Some of you don't know this, so go ahead and watch it if you don't know it. What are alleles, and what do we mean when we talk about allele frequencies? I'll talk about this a little bit in class too. He has a lecture, or 5 minutes, on Hardy-Weinberg equilibrium, and then some of the lessons that we can learn by using it. So I recommend watching these. I posted a couple of articles. In the homework, I refer to this paper, Wigginton 05. This paper gives the formula for computing the p-value for testing Hardy-Weinberg equilibrium. We'll talk through that probably on Wednesday, but if you want to go ahead and get started, look at this paper. I'll go ahead and show you the specific spot in the paper that you need. This is the formula that you're going to have to evaluate. So the probability of seeing a certain number of heterozygotes given the number of individuals and the number of homozygous recessive individuals can be computed, assuming Hardy-Weinberg equilibrium holds, assuming that there's no outside pressures. We can compute the probability of, in a population with this many homozygotes, heterozygotes, and homozygote recessives, if we let breeding happen, what is the probability distribution for the number of individuals that we would see who are heterozygotes? You can compute this, and then if you can compute that, you can ask, in my sample, we genotyped everybody because we did this SNP assay, so I know for each individual whether they're homozygous, heterozygous, or homozygous recessive. I can compare that probability from up here. I can compare this to my observed frequency of heterozygotes. And if my observed frequency differs greatly from what I think it should be under Hardy-Weinberg equilibrium, I should reject the assumption or the hypothesis of Hardy-Weinberg equilibrium. So that's a p-value that I need in order to make that decision. Do my data, are they consistent with the assumption of Hardy-Weinberg equilibrium? This is how we are going to compute the p-value. It's called an exact p-value. Notice that the p-value is an actual calculation based on the numbers that we saw on the previous page. That's exact in the sense that it's not an approximate p-value. When you do a t-test, 2-sample t-test, you have 10 observations control, 10 observations treatment. You assume the populations are normal. You do a t-test, you get a p-value. The p-value is, in that case, an approximate p-value. You're appealing to a large sample. Well, actually, you're not. You're appealing to the normality. But that is distinct from what's called an exact test. This is an exact test. If you're familiar with Fisher's exact test, that's exactly what we're doing. So again, we'll come back here probably on Wednesday and talk more about it. But if you want to get a head start on the homework, short story is you need to code this. A challenge with coding this, just FYI, is that all these exclamation points are factorials. So capital N is the number of individuals in our sample. So this could be thousands in a big study. Thousands factorial is going to break your computer. So if you tried to compute this as written, 2 to the this power times n factorial times this factorial times that factorial divided by all of this, probably going to have computational issues. The computer is going to say this is too big, that's too big, that's too big. So when I try to divide all these enormous numbers, there's going to be loss of accuracy. In the homework, I gave you a tip on how to get around that. Short story is compute these factorials on the log scale. So if I need to know, for example, what's n factorial divided by, what's 1,000 factorial divided by 900 factorial? If I say factorial 1,000 divided by factorial 900, I get a problem because factorial 1,000 is too big. It broke the computer. That's 1,000 times 999 times 998 times dot dot dot. What I can do instead is do it on the log scale using L factorial. So L factorial, if I do, let's try L factorial 1,000, 5912. So the log of the factorial is a number. When you log transform enormous numbers, they come much more reasonable. Factorial of 100 is enormous. Let's do factorial of 10. It's that. So I think if I do e to the L factorial, no. Yeah, I think that's right. E to the L factorial of 100. Yeah. Gave me what I wanted. So if, say, I wanted to do factorial 1,000 divided by factorial 900, like we tried here, what I could do instead is e to the L factorial 1,000. That's the log of the factorial. The log ratio is the difference of the logs. So I'm going to do this as L factorial 900, and I'm subtracting. So I still get an enormous number, but it could actually compute it. When you try to code the Wigginton formula up, see if you can use those log factorials. Otherwise, you won't be able to compute it. One other article, just real briefly, that is mostly just for a laugh. This was forwarded by one of my colleagues, Petsco 2012. So if you've ever been part of putting in a grant application, as a scientist, if you're going to go into academics or research, grants fund what you do. So you have to convince somebody, I know what I'm doing, I'm going to do this experiment, it's going to be wonderful for the following reasons, here's my budget, here's the risks, here's what could go wrong, here's my response to anything if it does go wrong, here's my plan B, you have to, all that stuff. And this is just kind of a fanciful, this was actually a peer-reviewed genome biology, it showed up in genome biology, but it's basically a joke. It's a conversation between some hypothetical people. So Spanish court in Madrid sometime in the late 15th century. Seated side by side are King Ferdinand and Queen Isabella. And before them standing is sea captain holding his hat in his hands. And he's here to propose a project to the king and queen. It's actually Columbus, that's right. Okay, so that's the funny part. It's Columbus. And he wants to sail around the world because he thinks that it can be done. It's not, you're not going to fall off the end of the planet like we believed at that time. So they debate and they worry and they say, you know, I have concerns about your grant proposal. Could you make such and such changes to make it more practical, more realistic, less risky? So Ferdinand and Isabella are talking about it. They turned him down and said, what else can we do? Let's see. Okay, here's another proposal. Let's look at another proposal. It's a completely new, big initiative, one that we can fund ourselves. It's a proposal from so-and-so to look for a very small change in the amount of water that one finds in cisterns. Accumulate as much data as possible and attempt to connect it with something interesting. We're going to collect lots of data, and we're going to try to find something interesting in the data. Columbus had a pie-in-the-sky, moonshot kind of project in mind. They didn't like it, but they do like this one. Nothing specific about what we're going to learn, but we're going to get lots of data. Surely there's something good. So what exactly are we going to find? He didn't say, just says that all the data will certainly tell us something. And then the joke is, oh yes, I remember that proposal now, the Gonsalvo Water Association Study, GWAS, catchy. That's real science. There is an issue, I guess, kind of a general issue nowadays with so much data that it's easy to fall into the trap of going on fishing expeditions in your data. I have all this data, surely there's something in here that can be useful. And as statistically savvy people, you know that if you get a data set and you look hard enough, look at it this way, look at it that way, this way, that way, you will find something. You know, if you look hard enough, the chances of finding spurious relationships goes up and up and up. So I think a valid critique of what we're doing with bioinformatics is this. We're getting lots of data. Surely this is going to be informative. But lots of data in and of itself doesn't guarantee valid conclusions. So anyway, just kind of a funny paper. The point of the paper really was just to poke fun at the grant getting process and how depressing it is. All right, let's go back to the paper that we had been looking at that goes over genome-wide association studies. And there's a couple of topics that I wanted to finish up with. So remember, GWAS is appropriate to common variation. So diseases that are common, that are thought to be driven by lots of small changes. So lots of places where there's variation, lots of SNPs, contributing to result in cardiovascular disease. Whereas we said something like cystic fibrosis is completely driven by just one change, which we call a mutation, because it's not common. High penetrance, high effect, low incidence. GWAS is not about that. That's an easier problem to solve. You just find a family that has cystic fibrosis and you follow them in sequence. The family members, you can find where the mutation is. Cardiovascular disease or something like that, we need lots of people because we need to talk about the population, not an individual family. So I need lots of people in my study, and I'm going to look at these SNPs. Okay. We didn't talk about this yet. So section 3.2, linkage disequilibrium. We have in humans millions of these SNPs, single locations where there's known to be fairly common variation among individuals. So we want to do one of these studies, association studies. Let's look at one of these really common diseases like cardiovascular disease, and let's try to figure out the genetic components that drive it. Well, there's millions of SNPs, and I need lots of people to do this. These types of studies, I need lots of people, thousands of people maybe. So cost is an important factor, even though next-gen sequencing is relatively cheap. I mean, much cheaper than what we could do not very long ago using Sanger sequencing and other things. Even still, this is a lot of SNPs to go genotype. And there's this idea that if you think about, we haven't gone into this in our lectures, but DNA, double helix structure, whenever mom and dad get together and decide to make a child, we get one of mom's chromosomes and one of dad's chromosomes. And they come together and they make my chromosome. I get DNA that has contributions from mom and dad. So mom has these genes, her own sequence of the human genome. Dad has his own sequence of the human genome. And the process of putting those together, what does happen is you get some of mom, some of dad. You don't get like mom complete and dad complete. So you're half mom, half dad. What really happens is you have a copy of mom, a copy of dad, and those are going to come together to form me. When they come together to form me, there can be switches among the two strands. So I could have some of mom next to some of dad. My genome doesn't look like mom plus dad. It looks more like mom shuffled with dad. So let's suppose we zoom in on one chromosome, one location of that chromosome. And on that location, maybe there's a few million bases on that chunk, and maybe there's a few hundred SNPs. Most of it does not change between me and other human beings, but there's a few locations, a few hundred maybe, that do change, SNPs. Okay, so we're going to go enroll 5,000 people, say, 2,500 who have cancer, 2,500 who don't have cancer, and we're going to genotype SNPs. In this little region, whenever mom and dad come together and start shuffling, things that, let me draw a picture. So if this is mom, that's dad, and there are genes in different locations along the genome. Actually, let's use different colors. So there's dad's version of these different genes. Whenever the combination event happens, things that are further away from each other are less likely to stick together. So if mom and dad come together to give rise to me, I may have mom's gene there, something like that. This and that are far away from each other. So if we're going to be shuffling pieces, like in this case, this piece came, followed by that piece, followed by this piece, followed by that piece. If we're going to start shuffling like this, then SNPs that are close to each other are going to tend to go with each other. Like here, those two genes are still together. And these two genes are still together. We could think of that as linkage. Those genes that we just highlighted are kind of linked together in the sense that they stayed together. So I inherited both of my dad's SNPs, versions of those, whatever SNPs are in those regions. Okay, so if we have millions of these SNPs across the genome, but I want to efficiently capture genotypic variation, maybe I can get away with not assaying or genotyping every SNP. In particular, if there's a region where there's 100 SNPs, and most of those SNPs being close together on the chromosome tend to go together, they're going to be highly correlated with each other. If an individual has this genotype at a SNP, it's probably going to have a predictable genotype at the SNP that's right next door to it. It's really close to it, because it's unlikely to have breaks, really fine breaks like this. I don't know if that makes sense. So the point is, if we want to be efficient, it would be useful to quantify these SNPs that we know of in terms of how related they are to their neighbors. And if there is a region where there's many SNPs that are highly correlated with each other, highly linked, maybe we could just take a few of them and get a sufficient picture of the variation at that location. Because all I want to do is associate. I want to associate cancer seems to have something to do with this area. And if there's 100 SNPs in there, maybe I only need 20 of them to find that it's in that area. If I do find that it's in that area and I did that trick where I only looked at 20 out of the 100, there is an issue because I don't know which SNP exactly it was. I know that that region is associated with cancer. So the next step might be to look more closely at that region. Now let's look at all 100 SNPs in this region and try to figure out the exact one that's associated. So linkage disequilibrium is a term that is relevant to trying to design one of these GWAS studies to require as few SNPs as possible. Linkage equilibrium, let me show you a picture. I don't want to look at that. Linkage equilibrium is what happens after a lot of generations. So after many, many generations, while SNPs that are close to each other will tend to stay together, if you have thousands and thousands of generations, eventually there's going to be breaks everywhere. Eventually, if you kept doing this, repeating it and repeating it and repeating it, for example, these guys are going to get split up. And those guys are going to get split up eventually. So depending on how old the population we are looking at is, linkage disequilibrium may look different. So for example, the authors mention the rate of LD decay is dependent on a variety of factors. And for example, African descent populations, they're the oldest. And because they've been subjected to these recombinations for so long, there's less areas of linkage disequilibrium. There's less of this correlation between nearby SNPs. Whereas Europeans and Asians, being younger, relatively younger populations, have more linkage disequilibrium. So if we're going to do this GWAS analysis and we're going to be clever about picking the SNPs to use, we may, or well, people do, design the studies specific to the population. So if you're going to do GWAS on African descent populations, you need more. You need a different set of SNPs potentially, more or less SNPs than if you were looking at Europeans. So if you look at GWAS data sets, you will often see, here's the experiment that we did. We have a few thousand individuals from this location of Africa. And then we have a few thousand locations of people, or people from Hong Kong. And we have a few thousand people from England. The reason we did that is to try to capture stories that are applicable to these different groups because they have different genetic characteristics. So briefly, linkage disequilibrium is something that we can assess. How correlated are two SNPs with each other? If you have 5,000 people in your data set, you can compute that. You can go get SNP1, SNP2, compute a correlation between their genotypes. And here are two ways that people typically compute this. This is basically what we are used to thinking of as a correlation. It's a square of a correlation, r squared. If r squared is really big between two SNPs, then we don't need both SNPs probably. So there's a famous project that went on 10 years ago, I guess, called the Hapmap Project, was all about this. Here's the millions of SNPs that we know of in humans, but using linkage disequilibrium to whittle that list down, the claim was that we can get more than 80% of the commonly occurring SNPs with 500,000 to 1 million on the assay. There's many more than 500,000 to 1 million. I don't remember how many there are. Anybody know? Humans? Okay. There's lots. So the Hapmap Project said, using this linkage disequilibrium idea, we can focus on a relatively reasonable subset of them. Indirect association is what I mentioned a second ago. Suppose once we do this filtering of the SNPs, we find a SNP in our data set that has a small p-value. It's associated with, say, cancer versus not cancer. The challenge now is that, well, that's not necessarily the magic SNP. It could have been one of the other SNPs that we used that SNP to kind of be a surrogate for, one of the other SNPs that it's linked to. Still, you could go look in the region at the neighboring SNPs in a subsequent study and try to drill down further. What else? Association testing, I've talked about this a little bit, but what we're going to be doing is a generalized linear model or linear regression. So basically just a generalized linear model. That could be linear regression, that could be logistic regression, depending on whether our response variable is a quantitative trait or categorical trait. Categorical, think case control study. You go get individuals who have cancer and individuals who don't have cancer. You genotype all of them at the SNPs, and you get a p-value for each of them. Are you associated with cancer versus normal? Multiple testing. Stephen, you asked about p-values and q-values a while back with GWAS. The SNP assoc package does not give you q-values or false discovery rates. It gives you, I'll show you when we have time, p-values. And it makes a picture of usually minus log p-value. Okay, so I think what these guys actually mention false discovery rates, which I think is the right approach. We're going to have 500,000 to 1 million SNPs. For each one of them, we're going to have a p-value. Based on what we've learned about multiple testing, the p-values themselves are limited in what they can tell us. They're not designed for that situation. That said, I think it is widely done in practice that people just look at p-values. I've seen that's what the SNP assoc function does. It gives you minus log 10 p-values. Problem with that is you don't know how small is small enough when you're just doing that. Because whereas if you compare or convert to FDR, you do. So I would recommend, if you're going to do this, to convert to FDRs. Take your p-values, convert them to FDRs. Okay, I think that's all I wanted to say on this paper. So next up, let's go see some R code. Let's see the Snips package. So I'm going back up to eCampus, scripts, and there is a gwas.r. All right. So need to install the package. I'm just getting BIOC Lite, and then I'll use BIOC Lite to install it. So once we get that in, I'm going to show you the data that you're going to play with in the homework. These are data, an example data set that came with Snips package. It's not a real-world size data set. It's kind of a toy example. Case control, so we have a categorical response. We only have 35 SNPs, so obviously not nearly as many as you'd have in practice. For each of the SNPs, we have genotypes for 157 people. For each individual, we also have some covariates. We have gender, arterial blood pressure, and something that is called protein levels, but I'm not sure what it actually means. Like we've seen with some of the other packages, step one, or before we can call the main workhorse function, we have to create a special class, object class, that contains all the data that the function wants. So we're going to do that right here. And then we're going to carry out an association test. Association test for a SNP can be done with the function association. We're going to do it for SNP1 right here. And all we're doing is logistic regression. So I didn't show how to match it here. I'll show you. If we have time, I'll show you right now. Otherwise, I will show you next time. Personal library. Yes. The use of the function association looks a lot like any other regression-based call in R. We say association response tilde covariates. For our data, once this gets loaded in, we can see it. There's a variable called caseCo, and that's the case control status. So these individuals are either cases or controls. They are, it's either disease or not disease, say. And we're going to model that in terms of gender, blood pressure, yeah. And I'm just going to look at the first SNP initially. All right, so let's see if this will work for us now. I'm going to run through here. That right there, data SNPs. SNPs is a data set that came from Snips. So when we say data SNPs, it loads that data set for us. Let's look at what documentation we have. Selected SNPs from a case control study. We're going to have two objects that come available to us. The Snips object is the data frame containing the actual genotypes. The other one tells us basically where each SNP is located, which chromosome and what position on the chromosome. Here's what the SNP data look like. So we have individuals in the rows. We have 157 individuals. I have a case co-variable, that's case control status. Let's look at how many of each we have. Table, SNPs, case co. So we have 110 cases, we have 47 controls. For each individual, we have gender, blood pressure, protein, whatever that is, and then all of our SNPs. SNP 1. For individual 1, chromosome 1 had a T. So this is one single location in the DNA. On chromosome 1 for this individual, remember there's two chromosomes. We're a diploid organism. I have two chromosomes of the same, like two copies of the same DNA. Chromosome 1, it was a T at this location. Chromosome 2, it was also a T. Individual 2 had the same genotype. Individual 3 had the same genotype. Individual 4 had a C on chromosome 1 and a T on chromosome 2, and so on. So what we want to do, let's just look at SNP 1. We want to ask, is genotype, whether you're TT or CT or CC, is that associated? Is there a significant relationship between that and the log odds of being a case adjusted for the covariates? Let's set up the SNPs, and then let's run the association function, look at its output, and then we'll close for the day. So this is the first thing I need to do, and what I'm doing is giving it the SNPs data object. I'm telling it where are the SNPs, which columns are the actual SNPs in. The first column is an ID variable, the second is case control status, and so on. The actual SNPs are in columns 6 through 40. I give it the info variable. Let's look at that one real quick. By the way, this is the other object that came, and it tells us that SNP 1 is on chromosome 1 at position 2,987,398. So start at the beginning of chromosome 1, go that many bases forward, that's the single letter that we're looking at. Let's do this. And then let's do this. That's the output of doing the association test. And we'll fill in some of the gaps of what all this is next time. For now, let me just say, when we ask for regression model like this, SNP 1001, what is that? It's not a number. It's basically a categorical variable. It could be either TT or CT or CC. So, and there's different choices of how to represent that categorical variable. So do I say, for example, that T is, or that C is dominant? If C is dominant, then homozygous C and heterozygous will exhibit the C phenotype. The only way you get the recessive phenotype is if you have homozygous T. Or it could be the other way around. C is recessive. So you get the T phenotype if you're either homozygous T or heterozygous. You only get the recessive phenotype if you're homozygous C. And there's a couple of other ones here. There's log additive, which says homozygous T is coded as 0. heterozygous is coded as 1 and homozygous C is coded as 2. So you've made it into a number. You could either treat genotype as a categorical variable or you could codify it. As I understand it, this is actually done a lot, the codifying. Do you know what in your world is typical here?
It depends. Like, for me, it's easy. I can just do it that way because I'm good at it. But most programs convert it to binary.
So 0, 1, 2 or 0, 1 or?
It depends. Like some programs you can do whatever you want, just say what it is, but like gap it to 0, 1, 2.
Okay. Yeah, I think. Okay, so yeah, I think this is pretty common. It's not a perfect choice. Okay. We'll talk more about it next time a little bit, but just be aware that this is something that we have to decide on. If I want a p-value for SNP1, are you associated with case control status? First, I have to tell R, how do I want to represent SNP genotype? Categorical, if it's categorical, who's dominant, who's recessive? Or do I want to do this numerical thing, calling it log additive? I think log additive is used a lot in practice because it's been shown to be pretty resilient, pretty robust. Okay, so we will finish there. Association does one at a time tests. There's another function called whole genome association that will go through and do a p-value for every SNP at once for you. These data down here, if you want to try it before next time, are more interesting. These are actual Hapmap data. There's 9,000 SNPs and 120 individuals. These are what comes above this association, WG association that I'm showing you with the toy data is really just to illustrate. If you really want to see the kind of output that you would get in practice, let's run this one. We'll run this one next time, but it takes a long time to run. Okay, that is all. Questions? Then I will see you Wednesday.